The central dogma of biology is really important. A dogma is just a principle. So basically we're just talking about the main principle of biology. And what this is it's just describing how we go from the gene in the DNA that's coding for the protein and how we end up making that protein. And it's just a two-step process. So first we're just going to transcribe or copy the gene in the DNA. And that copy we make of the DNA is called messenger RNA. And then we're going to translate or read the code on the messenger RNA and figure out what amino acids to put in what order to make our protein. And that's all the central dogma of biology is. All right, so looking at the two steps, transcription and translation, this is just a nice overview picture. So here's our double-stranded DNA here. And just remember that we've got our chromosome, our DNA molecule. And remember that each chromosome will have hundreds to maybe thousands of genes on it. And so here's one particular gene in our chromosome, maybe chromosome number 1. And then we will transcribe that gene, we'll make a copy of it, and here's our copy, our messenger RNA. And then we're going to translate the messenger RNA, we're going to read the code on it in our groupings of 3 and figure out what amino acid each of these, what we call codons, on the messenger RNA is coding for, and put them together to make our protein. Okay, it's also important to know where transcription and translation are occurring in our eukaryotic cells. So if we take a look at transcription, remember what we're doing is we're going to transcribe or copy the DNA to make our messenger RNA. So where we're going to be doing this in our eukaryotic cells, we've got to think about, well, where is the DNA located? So in our eukaryotic cells, the DNA is located in the nucleus. So that's where we're going to transcribe the DNA to make the messenger RNA. That's going to occur in the nucleus. Okay, so I would make sure you know where transcription is occurring, so that'll be in the nucleus because that's where the DNA is. And then translation, where we're going to read the code on the messenger RNA and make our protein, we need the help of the ribosomes. The ribosomes are little protein-making machines, and the ribosomes are actually out in the cytoplasm. They're not located in the nucleus, so they're out here in the cytoplasm. So translation is going to occur in the cytoplasm. So transcription and translation do not occur in the same place in our eukaryotic cells. So transcription will be in the nucleus, and then translation will be out here in the cytoplasm. So make sure you know that. Okay, the other thing is we are using both DNA and messenger RNA, so there are some differences between these two. We talked about the structure of DNA already. With RNA, with our messenger RNA, it's going to be single-stranded. We're going to use a different sugar, so instead of deoxyribose, we're going to use ribose, and that's why there's an R in RNA, and DNA uses the deoxyribose, and that's why there's a D in DNA. And the other big difference is that in RNA, we're going to be using U's, and in DNA, we've got T's. Okay, so that's another really big difference. And I know a lot of people forget that, and they want to put T's in their RNA, and there are no T's in RNA. There are U's in RNA instead. All right, so let's take a look at what happens in transcription. So we're going to be copying the DNA and making our messenger RNA. The enzyme that we're going to be using is called RNA polymerase. So if you just remember that RNA polymerase makes RNA. Okay, so hopefully that's easy to remember. Okay, so what we're going to be doing is we need to copy our DNA. Remember, the DNA is double-stranded, so we are going to be only copying one of the strands of the DNA. So we're going to be copying what we call the template strand. So we're going to be copying the template strand in our 3 to 5 prime direction, and then we're going to be synthesizing or making the messenger RNA in the opposite direction, in the 5 to 3 prime direction. And the DNA template strand and the messenger RNA will be complementary to each other. And that just means that our A's and U's and C's and G's will match up. Because remember, in the RNA, we don't have any T's, we have U's instead. So you can't say apple tree and chewing gum anymore. I haven't figured out a good saying for A's and U's, but you could still use chewing gum. Okay, so if I've got this DNA template strand here running 3 to 5 prime, let's fill in our messenger RNA strand that we're going to be making from the DNA template strand. So if I've got a T here in the DNA template strand, the messenger RNA would have an A, and then if I've got a C, I have a G. If I have a G, I have a C. And if I have an A in the DNA template strand, I would have a U in the messenger RNA. So don't put a T there, put a U. And then if I have a C, I'd have a G. T's, I have an A. And then an A in the DNA, I'd end up with a U there. Okay. So that would be how you transcribe the messenger RNA from the DNA template strand. All right, so the RNA polymerase enzyme, it is going to bind to the DNA at a special section on the DNA called the promoter. So the promoter sequence on the DNA just tells the RNA polymerase that, hey, that is the start of the gene. That's where you need to bind and start transcribing this gene. Okay, so the RNA polymerase enzyme will look for this promoter that just tells it that's the start of the gene, that's where I'm going to bind to the DNA and start transcription. Okay, and then it will transcribe the template strand, just one of the strands, all the way down until it gets to a certain sequence at the end of the gene that says, oh, that's the end of the gene. You can stop transcribing, and then the RNA polymerase will fall off the DNA. And then you can kind of see a close-up here on the left where you can see the RNA polymerase going along. It's separated the two DNA strands, and you can see that it's just transcribing one of the DNA strands, this bottom template strand in dark blue. And then you can see the messenger RNA that it's making in red here to match up with the DNA template strand. And so you can see that the messenger RNA, we're putting U's in instead of T's there. Okay, another idea that's you may not know about is in our messenger RNA, we have introns and exons. The introns actually do not code for anything, and they're going to lie in between the exons on the messenger RNA. So the exons actually do code for the amino acids. So the exons got their name for expressed sequences, and introns are intervening sequences. So once the messenger RNA gets transcribed before it gets translated, we're actually going to cut out the introns and then splice back together the exons. So here is on top here, we've got our messenger RNA that we've transcribed. So before we can go ahead and translate that, we need to cut out these introns, and then we're going to splice back together these exons, because remember the introns are not coding for any part of the protein, so we need to cut those out. So we do have things called spliceosomes in the nucleus that will go ahead and cut out the introns and then splice back together those exons. So once that's done, then we'll send out the messenger RNA to the cytoplasm, where the messenger RNA will then get translated.